Hello, everyone, and thank you for attending this GenScript-sponsored webinar titled Rapid Assembly of Gene Variant Libraries to Engineer Protein for User-Defined Goals. My name is Dr. Marinella Sandros, and I am the Senior Product Manager for Molecular Biology Services at GenScript, and I will be moderating today's webinar. It's my great pleasure to introduce you today to our speaker, Dr. Charlie Zhu. Dr. Zhu received his bachelor's degree in biotechnology from Nanjing Normal University, a master's in biology from St. Mary's University, and his PhD in microbiology and immunology from the University of Saskatchewan. His previous research focused on studying the Neisseria gonorrhea cell division interactome and rhizobacteria of legume crops and mechanisms of rotation benefit. Currently, he's a project manager at GenScript, assisting in project design and development for GenScript's library services. Now, to ensure this webinar has high sound quality, please activate listening only mode in your GoToWebinar settings or mute your microphone. If you have a question for the speaker, please type it into the chat box in the lower right corner of your screen for us to see. Dr. Zhu will be taking your questions at the end of the webinar. If we run out of time and he's unable to get to your question, we will email his response to you. Now, without further ado, let us welcome Dr. Charlie Zhu to present today's webinar, Rapid Assembly of Gene Variant Libraries to Engineer Protein for User-Defined Goals. Dr. Zhu, it's all yours.
Thank you, Marinella, for the introduction. And hi, everyone. This is Charlie Zhu from GenScript. I am the project manager for the evaluation of all your customized library requests. So today I'm going to give a talk about the rapid assembly of the gene variant libraries to engineer proteins for user-defined goals. Okay. Direct evolution has shaped the functions and properties of proteins found in nature, such that they contribute to the beneficial phenotype in living organisms. However, these functions and roles are just a fraction of those biologically possible. By modifying individual protein, scientists can go beyond what nature has evolved and gain completely new or at least enhanced functions of properties. Such enhanced proteins can be utilized to improve the phenotype of living organisms or have industrial or medical applications. The processes and frameworks for modifying protein sequences fall into the domain of protein engineering. There are quite a few downstream applications using such modified proteins, ranging from biocatalysis for food and industry to environmental, medical, and nanobiotechnological applications. Direct evolution employs an accelerated cycles of genetic mutagenesis and a subsequent isolation of improved mutants. This iterative process allows for the retrieval of genetic variants with improved performance for desired tasks and operational conditions. The general concept of directed evolution is very similar with the natural selection published by Charles Darwin. In direct evolution, sequence diversification and screening are often repeated multiple times to retrieve the candidate with the best performance. Each runoff cycle introduced additional amino acid substitutions and provides with a protein sequence closer to that of the protein engineering target. Direct evolution relies on methods from molecular biology to perform sequence diversification and methods from biochemistry, analytical chemistry, and microbiology to screen the resulting proteins for desired properties. Nevertheless, the entire screening process is usually laborious and less cost-effective. In some cases, proteins of interest are well understood in terms of its function and structure in nature. Thus, scientists often pursue a rational design rather than the directed evolution for protein engineering. Site-directed mutagenesis is one of the common tools to create desired mutants. Normally, there is only limited number of mutants generated using the rational design. This can significantly save the cost and time at the screening step. However, in most scenarios, one may have moderate level of background knowledge of their target proteins and may not be willing to spend extensive time and labor on screening assays. Therefore, needs for a balanced solution with less screening burden and information required for the target protein is growing rapidly. This is why we come up with our new semi-rational design. This is an interesting figure demonstrating the level of background knowledge of the protein of interest and a screening effort needed for different design strategies. Usually, the more knowledge we acquire about a target, the less effort will be needed for screening. A semi-rational design provides less pressure for screening purpose to our customers. The users do not need comprehensive knowledge about a target neither. Here in GenScript, with the acquisition of CustomArray, we can proudly employ the advanced semiconductor-based technology to produce single-stranded DNA for library construction. The custom oligonucleotide microarray is synthesized using a semiconductor-based electrochemical synthesis process. Each oligonucleotide probe is synthesized by a platinum electrode that is independently controlled by the synthesizer's computer. Synthesis is based on established phosphoramidite chemistry and occurs at thousands of sites simultaneously, according to a computer algorithm that activates only the specified electrodes. After the synthesis is complete, the oligos are removed from the surface and collected in a tube, creating oligo pools. The CustomArray oligo pool has several key features. Firstly, there are two types of microelectrode chips with different capacities for oligo synthesis. A 12K chip can synthesize up to 12,472 oligos at one time, with the diameter of each electrode in the chip about 44 micrometer, while the 92K chip has production capacity of 92,918 oligos. The diameter of the electrode in this chip is about 25 micrometer for each. So these two types of options allows us to choose different chips based on the expected variant library size. Secondly, since the number of oligos synthesized at each run is precisely monitored and controlled by the system, we are able to achieve a uniform distribution of variants in a mixed oligo pool. Thirdly, the system can synthesize all possibilities on a chip, and our library can always provide a minimum 90% coverage of all variants. Fourthly, unlike other chip-based systems, such as inkjet, we use a closed system for sequence synthesis. This helps maintain a sustained quality of our oligo library, often resulting in error rate as low as 0.5%, meaning one error in 200 bases without correction. At last, we can achieve the synthesis of oligo for library construction within a rapid turnaround time. For 12K chips, we can finish the synthesis as fast as 5 business days. Such a high-end technology has enabled us to pursue the next generation of library constructions with superior control of codon selection and distribution. Based on the downstream applications from our customers, we have further characterized the libraries into 3 different types. The first is the site saturation mutagenesis library, followed by the saturation scanning mutagenesis library as the second type. And third type is combinatorial mutagenesis library. Now I'm going to share a few cases with you about the quality we provide to our customers. And this is the first case. The first case is we're using the semiconductor-based technology. And the goal of this project was to generate a mutant library for an enzyme of interest to screen for a mutant with enhanced stability. The target protein is 404 amino acids in length, with limited information about itself and its homologs. So the customer wanted to test out all possibilities by having each position mutated to other 19 codon optimized amino acids, or in some cases, 20 optimized codon if the wild type is not optimized for E. coli expression. GenScript would deliver subpools containing all variants at each single position. Thus, the size of the library would be 7,760. And we guarantee a coverage rate over 99% as the QC standards for this request. To assure the final library contains as many as possible variants, we applied next-generation sequencing for this project. Next-generation sequencing verification was performed to calculate the coverage of our final deliverables. 7,759 variants were out of total 7,760 variants were detected. So there are nearly 100% coverage of all muted variants at each position, which are observed, and that matched with the customer's requirements. In addition, we have also tested the variant frequency using the data generated by NGS. The percentage on the y-axis on both figures represents the cumulative percentage of total amino acids. Each bar represents a mutated position, and each color represents the frequency of variants with a particular amino acid. We are able to achieve relatively equal distribution of amino acids at each position, indicating that there are no over or under representation of variants. Now it comes to the second case. The client wanted to mutate a total of 38 residues located at or adjacent to the active sites of an enzyme containing 484 amino acids. The goal is to obtain variant mutants with improved enzymatic performance selected using biochemical catalysis assays. These 38 residues were located in 3 clusters in the DNA sequence. For this project, the plan is to mutate each amino acid to other 19 possibilities at one time. There will be 720 variants in the mixed library that we deliver. Again, NGS is used for coverage verification and a codon distribution analysis. So our NGS results show that we can actually detect 722 variants in the final deliverables we deliver to our customers, meaning 100% coverage rate for all selected amino acids. And the distribution of each codon is relatively homogeneous in this library as well, which is consistent with the quality of our previous case. Now it comes to the third case. The third case is about antibody engineering. The customer was interested in improving the stability of an antibody candidate. Based on their bioinformatical analysis of the protein structure, the customer aimed to create simultaneous mutations for all 8 amino acids located at 2 clusters of the antibody sequence. Mutations at position 65, 66 in the first cluster, as well as 194 and 195 in the second cluster, need to be mutated to selected amino acids. Similar with the previous cases, this customer also asked for an even distribution of all variants. So NGS again is used in this case for verification of coverage and distribution. The figure on the left represents a theoretical distribution of codons at the position 194 to 197. On the right is the actual results about the scattering pattern determined by the NGS method. So as you can see, there were almost no difference at position 194, 195, and 196 between the design and the actual result. Some variation in distribution was present at position 197, but it is negligible to our customer. In addition, we have also performed an extra test to compare the quality of the libraries constructed using degenerated NNK mutagenesis approach to that of our newly developed chip-based method. Distribution of amino acids in degenerated library on the left is obviously biased at each position, while the codon distribution from the chip synthesized product on the right was homogeneous. Another cause of the degenerated library is that unwanted mutations, such as stop codons, will be inevitably introduced during the construction process, bringing additional screening burden to the users. Since our microelectrodes in the chip will only synthesize the required variants, this allows a precise control of the unwanted codons at the minimal level. Now, here comes with the service summary of precision mutant libraries offered by the GenScript. The site saturation mutagenesis library normally targets a consecutive or scattered amino acid region. Each variant in this library contains one mutation at a single position. All variants containing mutations at the same site will be prepared as one subpool for delivery. If the customer has preference for amino acids and their ratio at each position, we can actually adjust the mutagenesis range as well as the ratio for them. For scanning saturation mutagenesis library, mutations are normally introduced in a consecutive or a scattered region as well. In some cases, the entire DNA sequence are targeted for mutagenesis. Similar with the site saturation library, each variant in the library should contain only one mutation at a single position. But instead of delivering subpools for every single site, we prepare a pooled library containing all variants for delivery. At last, for combinatorial mutagenesis libraries, the biggest difference is that all targeted positions are to be mutated simultaneously, resulting in a very large size of the final library. And again, the pooled libraries are always the format of the final deliverables, considering the complexity of this type of library. Lastly, if the customer has the need for customized amino acid selections, or special request, racial request, we could always introduce them in this type of library. But evaluation is needed for additional requests. For all these three types of service, the NGS method can be applied to verify the distribution as well as the coverage in all three types of libraries.
Thank you, Dr. Zhu, for such a wonderful presentation. We will now move on to the Q&A portion of this webinar. Please take a few moments now to type your questions into the question box. While you are typing in your questions, I would like to introduce the advantages of GenScript's Precision Mutant Library services. GenScript Precision Mutant Library Services provides a perfect solution for cost-effective mutant library construction. Using our advanced semiconductor-based oligo synthesis platform and library construction provides precise control over amino acid distribution with equal representation of codons. The result is a well-designed library containing all requested mutations, preventing poor representation of variants and minimizing downstream screening burden. Our Precision Mutant Library services can facilitate a variety of applications, including determining critical residues of any gene or protein, improving structure of the protein, optimizing enzyme stability and substrate affinity, optimizing antibody affinity and engineering of CDR regions, creating a large number of variants for designing a metabolic pathway. GenScript can work with you to design and create your ideal mutant library to fit any project need. If you would like more information about our mutant library services, please visit our webpage listed here. In addition, GenScript offers a number of downstream services to complement your mutant library, including protein expression and antibody construction. For more information, please visit our website, www.genscript.com. All right, with that being said, we will start our Q&A session. Wow, we're getting a lot of questions. So here's the first question to you, Dr. Zhu. What is the timeline and price range for each type of libraries you mentioned in the slides?
So actually, the library construction can be highly customized. So effort that needs to be put into each case may vary a lot. The cost and timeline for each case need to be thoroughly validated by our team. So you can send us your request and our PhD level team will evaluate your request and provide you with the most cost-effective strategy with the shortest turnaround time. And if necessary, we would like to talk to you through a phone or online conferences.
Now here's another question, and they're asking, how are libraries supplied?
I think there are two different options for our customers. So if the customer wants to have the PCR products as the final deliverables, we can prepare the purified PCR amplicons in the liquid form, and the total amount will be dependent on the length of the PCR amplicons. But in general, we can provide up to a few hundred nanograms of PCR products to our customers. Alternatively, so if a clone library is the final deliverables, we will prepare about 10 micrograms of total plasmids in the liquid form, and we will also include 1 ml of glycerol stocks in the package as well. So in general, we recommend our customers to order the clone library from us because bias can be introduced during the cloning steps that ultimately will affect the desired distribution pattern in the plasmid libraries. And our professional technicians are trained to minimize the bias using our classified methods to ensure the quality of the clone library.
Now, do you have limits in library size? What is the maximum size GenScript is able to provide?
Yes, we do have the limits in the library size. For a clone library, the library size will be dependent on several aspects, including the copy number of the plasmids of your choice and the size of your final constructs. So generally, we're able to achieve a size of 10 to the 6th to 10 to the 7th using high copy number plasmids. So if necessary, we can scale it up to 10 to the 7th to 10 to the 8th. But that is the maximum we can reach by now. If the customer prefers the PCR products as the final deliverables, so the maximum size may reach 10 to the 11th.
Now, the other question we have is, why do I need rational diversity when I can screen a large number of random mutants anyway?
So there are a few numbers of the reason. Because you already know that certain amino acid substitution disturbed function of your protein. Like for example, the cysteines in the CDR regions of your antibody candidate. Or it could be because the number of possible variants of a protein is astronomically high, exceeding the capacity of even the highest thorough screening capabilities by many orders of magnitude. So the fewer useless mutations, such as those occurring in less important regions of the protein, or that cause frame shifts or stop codons, the better your chances of finding a variant that resulted in desired phenotype. And it can also because some screening assays are cost and labor intensive. So thus, screening fewer clones or save a lot of time and money for our customers.
Now here's another question. How are libraries supplied?
I think I have answered the question.
Sorry, I'm reading the same question.
Yeah, that's all right.
What percentage of coverage are you able to achieve?
Well, for our audience, you probably noticed that for the first case, we promised a coverage over 99% as the QC standards using the NGS. It was a very successful case that we had a real coverage of 99.987% as the result, with only one amino acid missed in the library. And in the second case, we could get a full coverage which showed a consistent life quality of our final deliverables. So both our customers are very satisfied with the high coverage we provided, and we are actually very proud of having a sophisticated system and an experienced production team in GenScript for library construction. So for future orders, we guarantee a minimum coverage of 90%. But in real cases, as you probably seen already, we can actually achieve a higher coverage that is close to 100%.
Now, what are the limitations in the combinatorial mutagenesis in terms of total number of sites and the number of consecutive mutations?
It is very hard to give an answer about the numbers in terms of the sites and the mutations. So the limit is actually dependent on several factors, including the scattering pattern of the mutation sites and the mutation pattern at each site as well. So we actually need to evaluate each case to have an accurate answers to this question. So please send us a request and we will evaluate based on a case by case and provide the most accurate evaluation to our customers.
Okay, so the last question we're going to be able to take today is, what information should I prepare when submitting the quote request for Precision Mutant Library?
Okay, so there are a few pieces of information I needed, or our team needed to evaluate, and also to shorten the turnaround time for communications between us and our customers. So, like first, we need to know whether you, as the customer, can provide a DNA template with a mutation pattern that you need for your library, and if so, we can just use your template for library constructions. And if not, we want the customer to provide at least the template sequence to us for evaluation. And as third piece would be, if a clone library is the final product, we need to know the backbone information. So we have about 153 IP-free vectors that can be used as the backbone for free in GenScript. So if the vector of choice is out of this 153 vectors, we need to know at least the vector sequence for evaluation. And if the clone library is the final deliverable, we want to know by the customer where is the insertion site on the backbone. So in some cases where the wild type sequence need to be replaced with the mutant sequences, then please indicate the exact range that you want to replace. And at last, sometimes our customers will have a special request, and we're greatly appreciative if you can provide any special request regarding the design of your library in the communication.
Well, unfortunately, we are out of time and have to conclude this webinar. Once again, responses to any questions that we were unable to answer during this webinar will be emailed to you directly. I want to thank you, Dr. Zhu, for sharing your research with us, and thank you to all who had taken the time to join us, and see you next time.
Thank you.